Welcome back to the course Fundamentals of General Immunology, and in the last class we started mucosal immunology, and in that we discussed about the gut environment and particularly about the immune system that does play an important role in gut homeostasis. We also looked into some of the lymphoid tissue which are present, such as galt, and also how this unique system of immunity in the gut can lead to tolerance. So today we'll try to discuss about the immune regulation, particularly in terms of mucosal immunity in lung or respiratory tract. So let's start with the discussion with our recent pandemic of SARS-CoV-2, which is COVID-19 virus infection. And what we generally know that the virus can spread very easily through the respiratory tract. And if we closely look into the virus, so the virus is roughly spherical in nature, and they have envelope, which we also call it membrane, and this membrane contains this very unique protein called spike protein, and after binding to the host cell with this spike protein, the virus does infection. Apart from that, we also know that the genome of this SARS-CoV-2 is positive sense RNA. So these are positive sense RNA genome, and that too without any segment. So these are single stretch of the genome. They don't have multiple segments in the genome. Now the virus can spread very easily in the environment, because once the infected person can sneeze, they can effectively produce more than 10,000 droplets, and if they can produce multiple droplets, the virus can remain in the air in the form of a suspension. And if they are in the form of a suspension, any susceptible individual which are coming in contact with those suspended particles can easily get inhalation of those virus particles, which will go inside the lung to start the infection. So apart from the actual biology of the virus, which is very critical in order to do an infection in lung, the spread of the virus is mostly through the respiratory route. So respiratory route is very obvious, very common passage for many different kind of allergen, dust, small or large particles, viruses, many bacteria, even lot of other important things which is spread through respiratory tract, simply because these are exposed to the external environment. We can inhale the air which are contaminated, and because of that region, this area become a constant source of infection. So let's try to see what is unique about the immune response towards the lung mucosal epithelium. So the lung mucosal immunity is again a specialized line of defense, similar like in gut. These are mostly covering the complete respiratory tract. We will try to see what are the parts of the respiratory tract. They also utilize similar like of mucus cilia like gut. So they have a lot of barriers which are present in the lining of the respiratory tract, which are full of ciliated epithelium. Also the mucus secretion is very high. We also looked into that there are epithelium which are called pseudostratified squamous epithelium, which are there in the lining of respiratory epithelium, and they can help to secrete multiple different kind of agents or kind of secretion which can neutralize the pathogen. In addition to that, we also see that there are huge amount of secretory immunoglobulins A that is secreted into the space around the respiratory tract. We have a very unique phagocytic cells of the lung called alveolar macrophages, and then we have a resident T cell population which are largely involved in memory, or also the immune response to the initial entrant pathogens. We also looked into that we have nasal associated lymphoid tissue or bronchial associated lymphoid tissue, NALT and BALT, which are largely comprising this lung mucosal immune response. So having said that, let's try to see the barrier which are there in the respiratory tract. So we definitely have a lumen, which is giving passage to the air, and of course this outer covering is full of mucus layer which are harboring bacteria as well, because whatever we inhale is not sterile. Most of the time we are inhaling a lot of dust, bacteria, other pathogen. Most of the time these are all harmless, but sometimes it could be harmful, because once the immune response is suppressed or due to some infection or some other disease biology, we can see that some of the opportunistic pathogen can start doing the infection. Apart from that, we have antimicrobial peptide and the secretory immunoglobulin A, that's in the dimeric form, may be present in this mucosal inner side of the epithelial, touching the epithelial surface. Of course, the baseline epithelial will also always be there, which is having a ciliated epithelium. Now, so these are the structure, typically a structure of a mucosal barrier. Similarly, in the gut also, similar in the intestine also, similarly in the genitourinary tract also, we can see similar kind of pattern where you have a lumen. Apart from that, we also have a mucus secretion, and then we have a lot of secretory immunoglobulins or antimicrobial peptides touching the lining of the epithelial cells. So when we see the anatomy of the respiratory tract, when we say anatomy of the respiratory tract, which stands for what are the different parts of the respiratory tract. So it starts with the nasal cavity, which is a hollow cavity which takes the large amount of air, which then terminates into a small narrow part called pharynx, and this pharynx is actually terminating to another smaller part called larynx, which terminates into trachea. So these are the cartilaginous rounding ring-like structure, and then finally this trachea goes to this structure which are entering inside the lung called bronchi, and this bronchi is again branched into small bronchioles, and finally the small kind of unit which are the actual functional unit, which is called alveoli. That's the last unit of this respiratory tract. So this is actually actual respiratory zone where we see the exchange of gases from the atmosphere that's happening. So we have carbon dioxide which is going out from blood cells to the alveolar space, and also vice versa. So essentially we see these alveolar spaces are involved in gaseous exchange, carbon dioxide and oxygen. So trachea, bronchi and bronchiole, in general these are called as lower respiratory tract, while nasal cavity, pharynx and larynx, these are in the upper side, therefore they are called upper respiratory tract in general. Now these are generally the part of the respiratory system, and if we see, these are constantly under the pressure of the external contact, and therefore we need a separate or a specialized kind of immune system that can control anything which is entering through this route. So, and that is why this whole anatomical reason is important. Now let's try to understand the surface epithelium of particularly the respiratory tract. So these are actually large airways, which are large size airways, and then we have a small size airways. We can consider this as a bronchioles, and this could be in the part of the lower, the airway which is touching the alveolar epithelium. So in the lower respiratory epithelium, we can see that there are multiple different kinds of cells which are present. Some of the well characterized cell is already known to us. One is goblet cells which are secreting mucus. Then we have an ionocytes which are actually responsible for electrolyte balance, so which is written here. So these are spatially ionocyte, which are mitochondrial rich cells, and they maintain the electrolytes and also acid base balance, and they can mainly involve in transportation of sodium, chloride, H plus ion and also bicarbonate ions. So these are important ion transporter of the lung epithelium. Then we have a ciliated cells which are actually the cells of the respiratory tract, and then we also have M cell, that's called microfold cell of the immune system, that can useful while entrapping the pathogen from outside and then transferring into the Peyer's patches or and then transferring into the lower epithelial areas. If we see the small airway instead of a larger duct, if we look into what is there in the smaller duct, smaller changes in the duct, we see that this could be a basal cell which are there in the cellular system. We of course have M cell, club cell. It's also called as cholera cell, and these are specially secreting some of the very important proteins such as CC16 or CC10. And then we have a tuft cell as well in this lining. The tuft cell are actually chemosensory cells, and these are involved in fighting with the parasitic infection and some of the stimuli which are coming from outside. And also there is a report that says that these are secreting interleukin 2 and also act as a stem cell to do regeneration of the epithelial surface. Apart from that, we also have a lot of neuroendocrine cells which are present, which are involved in secretion of different hormones in the respiratory epithelium. So these are typically the pattern of lower respiratory epithelium, and when we say lower epithelium, these are divided into the large duct and the small duct, and therefore we call it large airway and the small airway epithelial surface. Now let's try to move on and see all and some more details about how lung fights with an pathogen. So once there is a detection of a pathogen in the lung epithelium, so dendritic cells which are present, or the phagocytic cells which are present in the lung interstitial space, can engulf and process this antigen, and further processing they can migrate to the nearest lymph node. And once they go to the lymph node, the normal processing of the immune cells such as the B cell and the T cell can take place in that particular area. They can also secrete a lot of cytokines, and then the cell proliferation and cell differentiation can also happen in the lymph node. And further, they can come to clear the infection again to the lung epithelium. So they can try to clear the infection. Some of the cells are involved in eyeball formation, which is called inducible bronchus associated lymphoid tissue, where they can do a lot of other processes. And then finally, they can also go into RAMD, which is called repair associated memory deposits, and these are the resident memory T cell accumulate and self-renew in case of reinfection. So these are actually the cells which are there in the lung that can fight to the infection and act as a reservoir of a memory cell. So these are very unique way the lung can tackle the pathogen entering into the system. So once the pathogen is entering, they can be identified by phagocytic cells, and these phagocytic cells, once sensitized, can go to the nearest lymph node, and from lymph node they can channelize the B and T cell population, which again go to the lung and try to clear the infection by producing a lot of cytokines and other kind of chemokines. So this in general the term that takes place when we are talking about an active immune response in the lung epithelium. Apart from that, there are many other things which we need to understand about the lung epithelium, and some of the things which are pretty well known in the field is when we old, the lung mucosa environment is getting senescent, and because of this senescent environment, the people may be susceptible for more viral infection or more kind of disease problems. So in general, the elderly people are getting more prone to the infection, particularly in the respiratory epithelium, because of the change in the structure of the epithelial area. And some of the change in the epithelial surface area are listed here. For example, in the healthy alveoli, we see that there are two different kind of pneumocytes. One is called pneumocyte type 1 and the other is called type 2. These two pneumocytes are having different function. The type 1 pneumocytes are mostly involved in secretions, and type 2 pneumocytes are also involved in secretion. Apart from that, they can also do a lot of processing, including the surfactant production and mucus secretion. Apart from that, we also have the lining of the alveoli filled with some fluid which is called alveolar lining fluid, ALF. Once there is a senescence, when the person is particularly approached towards the 40-45 then age group, then we see that the pneumocytes are getting older and their functionality is also getting diminished. And because of that region, we see that the ALF is getting oxidized. Now why it is happening, that once there is an aging process, lot of reactive oxygen species and other things are coming into picture. So here we have just listed it out. Under normal condition, like in healthy condition, the alveolar lining fluid contains very high amount of antioxidants such as glutathione. So glutathione is an antioxidant which can remove the reactive oxygen species which are generated as a toxicant in the human body. So once there is a senescent cell, or the cells are not functioning properly, we see that the reactive oxygen species concentrations are getting increased, and because of this region we call this whole area to be oxidized in nature. And what we have, the fluid, earlier it was very useful, now become an oxidized form full of oxygen radicals and other things. And therefore we see that when the young, and when we compare the disease probability, the young ones can fight the disease very effectively, simply because the active cells such as macrophages, neutrophils, and the secretory immunoglobulin cells such as B cell are still active, while in case of aged individuals, the pneumocytes are getting, say, not functioning properly, and also there is a dysfunctioning of the immune cells, and therefore the alveolar area or the complete respiratory tract are susceptible for various infections. So this is one such point which we need to critically look into when we discussed about the old people, why old people are become more susceptible for disease conditions, simply because of the change in the cellular architect of the lung epithelium surfaces. Now let's try to look into some of the disease conditions which are very critical when it comes to lung. So one such disease is called chronic obstructive pulmonary disease, COPD, very common form of disease in cities nowadays. So chronic stands for long term. So chronic means any infection which is going very long lasting. Opposite to that we call acute, so which is very fast in nature, but here we are talking about chronic, which is a long lasting, prolonged duration. Obstructive means something which is stopping the flow. Pulmonary stands for something which is related to lungs, and then disease is just a condition where there is a discomfort. So the disease is called COPD, and that's happen in case of individual which are constantly under the exposure of some of the noxious substances such as smoke from the cigarette or industrial smoke, or say some kind of pollutants which are coming and not suitable for the human being. So these exposure can cause the environment to be susceptible for infections such as bacteria, a small dust particle or viruses can easily come into the site and could lead to upregulation of eosinophil, macrophages and neutrophil. So essentially these things are coming into lung epithelium, and lung become sensitized to produce more of the inflammatory cells and the immune cells, which in turn can produce a lot of different cytokines, which we are not listing many of them, but just try to understand here IL-1 beta, which is a pro-inflammatory cytokine. Reactive oxygen species, we just now discussed about why these reactive oxygen species are getting an environment oxidized and causing a person more prone for infection. TNF alpha also a very important factor which are secreted by these immune cells, and these collectively can cause a lot of problems in the lung. One such condition is called emphysema. The second is called small airway disease condition, which could be many other things, and otherwise they can cause bronchitis. So bronchitis stands for the inflammation of bronchi, which is the inflammation of this particular area. Emphysema stands for the mixing of the alveolar space into a big hole-like structure. So the alveolar are the small area which are giving a large surface area for the gaseous exchange. So these are small kind of a sac-like structure in the lower part of the lung area, and once these emphysema-like condition happen, then essentially these all structures are getting fused, and this become a big large sac which reduce the surface area of the lung epithelial, and therefore the oxygen carrying capacity is also reduced to those persons, and this condition is what we call emphysema. It's a very common condition in chain smoker, cigarette smoker, where the lung epithelial become merged to form a big sac-like structure where the oxygen carrying capacity of those individuals are getting reduced. This is what we call, condition is called emphysema. So collectively the disease condition is called chronic obstructive pulmonary disease conditions. So let's try to move on and see some other disease condition, and one such thing is the allergic condition which is happening in the airway epithelium. We just now discussed about the smoke and other thing which can lead to chronic obstructive problems in the lung. The allergen can also cause the secretion of multiple cytokines such as IL-33, IL-25, or also thymic stromal lymphopoietin. These are very specific cytokines secreted from the lung epithelium. Apart from that, these allergen can bind to the surface and can activate the dendritic cell to process those allergen for the CD4 positive T cell population. That means there could be a possibility that the cells intestine, there could be a possibility where the lung epithelial cells can be infected, or say coming in contact with those allergen can produce different kind of cytokines, and these cytokines can percolate down to produce a lot of different, say, activation pathways. One of them could be the activation of innate lymphoid cell. So the innate lymphoid cell can be activated, or else they can also activate the CD4 positive T cell, which otherwise can be activated by processing the allergen through dendritic cell. The dendritic cell can present those allergen with the MSC molecules, and these T cell receptor can recognize those MSC2 molecules, and then can sensitize those cells to produce some of the cytokine by class switching phenomena, and one such cytokine is IL-4 and IL-5. So IL-4 and 5s are specifically responsible for allergic reaction, while IL-5 is also known to activate the eosinophil molecules, and of course IL-4 can do a lot of activation of B cell and can fight with the allergens. Apart from that, the innate lymphoid cell can also activate and produce IL-13, which can go to the site of mucous epithelium in order to nullify those all entering allergen, or try to subside the infection or subside the issues which are generated by the entry of those allergen. So allergic airway inflammation is mostly related to the secretion of IL-4 and IL-5. So remember, IL-4 and IL-5 is actually a very important cytokine that does play a role in allergic infection. So these are some of the problems which are related to lung epithelial areas. Let's try to move on and see some more important thing. Of course, we'll try to learn some of the very important aspects of SARS coronavirus 2 infection in lungs as well. So there is a condition which is called cystic fibrosis. Now cystic fibrosis is a hereditary disorder where the protein called CFTR, which is called cystic fibrosis transmembrane conductance regulator, CFTR, is a protein which is present in lung epithelium. And also the gene which are getting mutated may not produce a functional CFTR, and because of non-functional CFTR we see a lot of problem, and the collective those conditions are called cystic fibrosis. Now let's try to focus here and see what is happening in the normal or healthy lung. So when the lung is healthy, the passage is very clean. The channel called CFTR is a chloride channel. So this is an ABC transporter protein. When we call ABC transporter, that stands for ATP binding acids. So they can utilize the energy and try to push out the ions into the extracellular space. So when the chloride ions are, say, more in the inner area, these chloride ion channels are getting opened and the chloride are flushed out. To balance the chloride ions, the sodium are getting in with the help of this epithelial sodium channels, and that is what happening all the time in the lung epithelium to balance the NaCl concentration in the epithelial lining. So once there is a functional CFTR, the chloride ion is getting pumped out and the sodium is getting pumped in by this sodium channel. So these channels are very important to maintain the homeostasis of the cellular system. What happens when there is a mutation in the CFTR protein? So suppose if the CFTR protein is getting mutated, it's not getting a functional protein, or for example, if there is a deviation in the functionality of the protein in terms of they are not binding properly to the chloride ion, or if their concentration itself is less in some individual, then we see that chlorides are getting accumulated in the inner side of the epithelial area. That means chloride is getting accumulated more and more into the cellular space, and to control that, more and more sodium ions are also coming into internal space, and therefore the water is getting absorbed into the cellular system, making the mucus very, very thick. Now what is happening here, the mucus is actually very fluidic in nature because of the water concentration. So since the CFTR is not functioning in a proper way, the protein is blocked, and therefore most of the chloride ions are just stuck inside. To maintain the homeostasis of the chloride, the sodium channel is also opening to a large extent in order to make more concentration of sodium in the inner side. And since more sodium and chlorides are coming in the internal side, water is also oozed out from the mucus layer to the cellular side in order to maintain the homeostasis, because otherwise the osmotic imbalance will cause the cells to burst out. And just to stop the cellular death, the cell can maintain the homeostasis and the water is coming inside the cellular area. Now because of these all changes in the dynamics of the sodium chloride and water, the mucus becomes very, very thick. And therefore once it is thick, we can see that the pipe which is carrying the air is full of loaded viscous fluid, and therefore the person is unable to breathe properly because the air is getting obstructed into these passages. And once there is a passage obstruction, we need to really take out some physical ways to get the mucus out from the individual, otherwise the person may have a suffocation or unable to breathe properly. There could be a possibility in the young individual where there is a mutation, they used to tap their chest in order to get the mucus out. So there is some physical agitation required in order to remove the mucus from that. So the CFTR is actually the culprit, the gene where the mutation can cause this problem. But again, this whole pathology is related to lung epithelium. Therefore, this cystic fibrosis is a target of a gene therapy. People are trying to rectify or correct the CFTR gene to send it to the person who is actually suffering from the disease. And once the person is corrected for the CFTR gene, they can produce a functional gene and they can start their lungs activity in a proper way. So this is again a lung-based pathology, but again it all depends upon the immune response, which is targeted towards a very specific ion channels. So apart from mucous immune response, we also need to make a homeostasis of the ion channel as well in the lung epithelium. Let's move on to our final discussion of this lecture, SARS coronavirus 2 disease. Now we know that SARS coronavirus can cause various degree of lung pathology. The lung could be very healthy when the alveolar areas are intact, having a very high surface area, and then we can see the exchange of gases. While in infected condition, the lungs alveoli can be moderately damaged or severely damaged. It all depends upon how much fluid is accumulated in the alveolar space. If the fluid is completely occupied, then there is no space to trap the air, and therefore the person may have an oxygen deficiency and they can have a severe respiratory passage obstruction. And if the person is having a moderate blockage of the alveoli, then there could be a chance that they can still do breathing and the exchange of gases to some extent. But again, we need to do an immediate treatment to those individuals where these problems are happening. Now, what is happening in case of moderate damage? Now, in moderate damage, the inflammatory signals are coming into the space. We can see that the small volume of the fluid is also accumulated in this area. The infected alveolar cells will produce different kind of cytokines, and also the area, because of this cytokine, we can see that the area become dilated out. and therefore we see vasodilation can happen in the blood vessels. So this is an alveolar area which is rich in supply of the blood vessel. Now what is happening is the oxygen can go inside the blood vessels to do an oxygenation, and the impure blood can pass on the CO2 into the lung alveolar, and therefore the exchange can take place. So this is a very important area where the oxygen and CO2 exchange can happen, and therefore we need a free space in order to do these processes. And if that's not happening, like in case of severe damage of alveoli, we see that the whole lung alveoli is full of fluid, and also the interstitial space. Interstitial space stands for the space between the alveoli and the blood vessels. These are also occupied by the fluid. In addition to fluid, we also see that the scar formation can also happen because of the damage in the lining of the alveoli and also the blood vessel. Lot of inflammatory cells such as neutrophils and other kind of things are coming into picture, and they can produce multiple cytokines, and this can again exaggerate the disease condition. Also the surfactant which can give the fluidity of the space is also lost in due course of time, simply because there is no exchange of the gases are happening, and of course exchange of gases are highly compromised simply because the area is full of fluid. So this is what is happening in the coronavirus disease condition. Not in coronavirus, but most of the respiratory infections are happening in this way, while this is called severe acute respiratory syndrome because it's a collection of disease. Syndrome is something which is a mixed form of many other disease conditions. So the collection of all diseases is what we call syndrome. So let's try to understand the immune response of the SARS coronavirus 2 infection. So now there are two terms which we need to understand. One is called angiotensin converting enzyme 2. Angiotensin converting enzyme 2 is actually a receptor. So the SARS coronavirus 2 can bind to the AAC2 receptor which are present in the lung epithelium, and after binding to the receptor, the virus can enter through the receptor mediated endocytosis, and once they can engulf by receptor mediated endocytosis, then we have some sensors available in the endosome called TLR3 and TLR7. So these are some of the innate immune sensors or toll-like receptor which we have discussed in the earlier classes as well. And apart from that, these genomic RNA can also bind to MAVs, which stands for mitochondrial antiviral signaling proteins, which are located in mitochondria, as name itself suggests that these are associated with antiviral response and also associated with mitochondria. So they can also sense the genomic RNA of the coronavirus, and once they are sensitized either with the MAVs or the toll-like receptor 3 and 7, they can activate the NF-kappa beta and IRF pathway, which in turn will go inside the nucleus. NF-kappa beta will produce a lot of cytokines, and IF can produce a lot of interferons. So these can produce cytokines and lot of interferons, and these can pass on to the neighboring cell population. So these type 1, type 3 interferons, type 2 interferons, inflammatory cytokines can sensitize the dendritic cell and also activate CD8 specific T cell immune response, which entirely do into cumulative effect. And finally, these T cell population, cytokine and other things come into sight in order to do induced apoptosis to the epithelial cell, and therefore they can produce an antiviral state in the lung epithelium. So what we understood till now is lung is a very fragile organ, and why it is very susceptible, because they are in constantly touch with the external environment. They are constantly getting air contaminated, dust particle contaminated, different size particles which are directly entering inside the lung epithelial area, and lung is such a site which are directly giving an excess of the blood vessels exchange. So this lung alveoli can directly come in contact with the alveolar space with the blood vessels to do an exchange of oxygen and carbon dioxide, and therefore these are very susceptible areas that are prone for infection. Some of the conditions which we discussed today is COPD, the chronic obstructive pulmonary disease condition. Then we also looked into some allergic condition where IL-4 and IL-5 does play a very important role. We also looked into why the older people are having susceptibility for the disease condition, simply because the lung epithelium is not functioning properly, and there could be more and more accumulation of reactive oxygen species. And also we looked into the SARS coronavirus 2 pathology, how the disease can cause obstruction and obstruct the passage of gaseous exchange at the level of alveoli and the blood vessels, and also a little bit of the SARS coronavirus 2 immune response. So this is all about the lung-based immune response, and in the next class we'll try to understand about the genitourinary tract and some of the unique thing about the genital and the urinary tract immune response. Again, those are all mucosal epithelium, because they are always moist and produce huge amount of mucus, which are giving susceptibility for many infection, because they can have a very rich protein source that can give say opportunity to many bacteria population to survive there.